Okay, hey there, biochemistry students. This is the week 5 and 6 video. And sorry that it's a little bit overdue, but you'll note that there was no week 4 video because you all were working diligently on your assessments and your labs, and I have been working diligently to give you a lot of feedback on all the competencies associated with those so that you can improve as the year goes on. Remember that the actual numerical grades don't really matter this early on. And so this is really about giving you feedback. So been trying to prioritize that. And the downside to that is that then I don't have time to record videos like this. So thanks for bearing with me while I got through all of your labs and started in on the assessments. And now I'm taking a break to record this video and also the next one. So this video will span the last couple of weeks of the class. Technically, I'm recording it at the beginning of week 7 when we're starting to talk about cell organelles, and I will record a follow-up video next that's for week 7 and 8 and will be about cell organelles. Okay, so you have that to look forward to. Now, just to refresh, because this will therefore be sort of review by the time you're watching it, to refresh your memory on what we did to transition from the first unit to the second. I emphasized to you that we have to look at molecules, which we learned about in the first unit. We learned that they're made of atoms that are covalently bonded together, and that mostly with living things, it's P, phosphorus, H, hydrogen, O, oxygen, N, nitrogen, and C, carbon that are involved in these covalent bonds to make molecules in living things. We have to put molecules into their context as either being in a cell, cells being the unit that living things are made of. So they're either in the cell, acting as part of the cell, in some way helping the cell to carry out its functions and stay alive, or they are outside of the cell. And that would be called an extracellular environment, extra meaning outside of. So extracellular molecules might include molecules that are helpful to an organism, and the organism might want to bring them into its cells so it can use them. Like if food is in the environment, you'll want to bring that in to your cells in order to use it, right? But also something that you find in extracellular environments is, pollutants, things that are potentially deleterious or problematic, things that have damaging impacts if they find their way into cells. And we read the story of 6-PPD. You see the molecular structure here in the top left of this slide. 6-PPD is a material that is in tires that you find on automobiles. It is a molecule that is added to them. It is synthetic, which means that it doesn't occur in nature. Humans make it in a laboratory. And they add it to the tires in order to make the tires, this is kind of ironic, in order to make the tires wear down more slowly. So this molecule's purpose is actually to help the tires to not wear down as quickly. The downside is that when the tires do wear down, the 6-PPD is one of the molecules that flakes off of them. And not in individual molecules, but in little chunks of rubber that contain it. So you'd find this kind of stuck in the little chunks of rubber that are flaking off of this tire. Those are microparticle pollution that can wash into our waterways. And I did this reaction for you in class. I built 6-PPD, the structure, and I talked about how when it's exposed to ozone, ozone can add oxygens at a certain section of the molecule, this middle ring here, and that turns it into a slightly different molecule that research suggests is lethally toxic to at least some species of fish. In fact, coho salmon were kind of what the article led with as a species of fish that tends to experience huge die-offs when the first rainstorm hits during our winter here in the Bay. And in fact, that specifically has been noticed in the South Bay, where we go to school and where many of us live. So the fish die-offs, it was determined, are likely due to the presence of this chemical 6-PPD quinone, which gets into their cells. And so the theme of week 5 was kind of like, why does this molecule get into their cells? What even makes the cell separate from the extracellular environment? And why would this molecule in particular go into cells easily? Well, let's review that, shall we? Cell membranes are made of phospholipids. Phospholipids, you can see on the right here, are made of a head that is hydrophilic Oops, sorry about that. A head that is hydrophilic, meaning that it is water soluble. Water soluble. And another way to think about that is it must therefore be polar. And to refresh your memory on that, polar describes a molecule where the covalent bonds are uneven. By uneven, I mean that one of the atoms is pulling the electrons that the bond is made of. Every bond is 2 electrons. It's pulling the electrons toward itself and making itself a little bit negative, and it's pulling them away from the other atom in the bond and making it a little bit positive. And that creates the situation that makes water do hydrogen bonding, meaning that one water molecule can stick to another one. See how I'm reviewing this concept because it's really important. One water molecule can stick to another one like that. Okay, partial positive hydrogen sticking to a partial positive oxygen from a different water molecule. And this hydrophilic head, you'll notice, has lots of oxygens involved, which render it polar. And that makes it also have partial positives and partial negatives. The phosphate group actually has full negative charges. You don't need to worry about the details of that or this like extra group that gets added on here. Those are things that are beyond our concern at this juncture. If you take further advanced biology classes, you might learn about those. But for now, our understanding is basically this is a charged area that can interact with water in the same way that water hydrogen bonds with itself. And that makes it water soluble. It can dissolve in water and interact with water. Hanging down from that head group are 2 commonly called tails. Okay, these are hydrophobic tails. And those hydrophobic tails are hydrophobic because they do not contain polar bonds. All of the bonds in those tails are non-polar bonds. Okay, that would make them not water soluble, but fat soluble. Okay, and you can see these are actually made from fats. Okay, these chains that hang down are actually made from the fats that you eat. So the fats that you eat, one place they go is to your cell membranes. Okay, so fat is good, especially healthy fats like olive oil and fish oil and flax oil and different avocado oil, vegetable oils for the most part. Although corn and canola are so commonly eaten by Americans that we probably don't need more of those. But yeah, other ones are generally healthful because they add a variety of fatty acids, these fats, to the phospholipids in your cell membranes, and they keep your cell membranes functional. So fat is good, just not a crazy huge amount, that's all. And try to have variety. Anyway, this is another way of drawing the phospholipid. And when a bunch of them form a layer, they do it like this, meaning that a cell membrane basically looks like this here. So this could be outside of the cell, and on the other side of the membrane, this could be inside the cell. Therefore, getting across this membrane means going from outside to inside, or of course, going from inside to outside too, which is an important concept. Cells sometimes want to get things out. And so we'll talk about that as well. Okay. So now let's look at how things move across the membrane. Basically, there are a couple of different possibilities. Either, and you'll recall from our membrane lab, either they can move right through the membrane. Okay. This would be if they're like small and they're non-polar, and we'll talk more about that in a minute. So some things can just diffuse right across the membrane, no problem. But many things need some kind of tunnel. In our membrane lab, we built a tunnel out of a straw chunk that you coated in membrane and you stuck it in the membrane. Then you were able to pass something through like it was a tunnel without, breaking the membrane. So there are different types of tunnels you can put in there. You can see there's like selective diffusion where only certain types of molecules are allowed in, or there's just like facilitated diffusion where it's just kind of opens up and whatever comes in. Don't worry about different types of diffusion for now. I chose this image because it was the best one I could find of like the protein channels. Just recall that these are protein channels. These orange and purple molecules. They're made of proteins. And they are the tunnels that allow certain molecules to move through the membrane. Okay, so first key concept, some molecules can slide right through and other molecules need a tunnel to get in. Follow-up concept is there's another way to get things to go across the membrane, and that is to coat them in membrane, to actually put a bubble of membrane around them. This is like when I asked you to put your hand through the bubble, and you all were able to do it. If you first got your hand totally coated in the soap mixture, then you could put it right into the bubble without popping it, which is pretty fun. Similar deal with a cell. If you have a bubble of membrane, and we've learned the name for that now, it's called a vesicle. That's a bubble of membrane that can transport things around. Well, you can put the stuff you want to move across the membrane inside of a bubble of membrane, and the bubble of membrane can then move toward the cell membrane. And when it bumps right up against it, it can actually fuse with it. It actually, like, the bubble merges with the cell membrane, and the cargo that's inside can then come out and go into the cell. And again, you could also run this in reverse, like if this is outside of the cell and this is inside of the cell, then this would be a vesicle that is dumping its cargo into the cell. You can also go the opposite direction. You can go from inside to outside, the exact same mechanism. Vesicle, which is a bubble of membrane, fuses with the cell membrane and dumps its cargo, and you could do it outside, from inside to outside. Okay. Now, the final kind of follow-up question here is which type of molecule will undergo which type of transports. And here, this is a little bit of a busy diagram, but the key thing to note is that at the top, the first thing of real importance is it small or is it large? Okay. If it is large, then you need a vesicle of some sort. Okay. You need some kind of coating of membrane. The examples of large molecules might be like, large food items, for example, or like a large polymer, some kind of really long protein that just is not going to fit through a tunnel. Okay. So the idea here is that if the cargo is big enough, then there's no way to put it through a tunnel. You might think of this as like, if you have to move a really large, let's say like a wind turbine blade or something, in reality, those are really hard to ship around because they're actually so massive that just getting them from the place that they're manufactured to the place where the wind turbine is going to be built is really difficult. And one thing you probably avoid is going through tunnels because they're simply too big. They have to go on these giant trucks and they have to be accompanied by an escort, of multiple cars to make it safe to move them around. So with really large cargo, as in this analogy I'm making, the key would be to put them in a vesicle and then fuse the vesicle with the membrane. You'll notice that there are like other fancy options here. And for now, I'm going to avoid worrying about these processes. We may come back to these if and when it makes sense, but right now I don't want to overwhelm you. So we're just going to focus on the concept that if you coat something in membrane, it can fuse with the cell membrane and it can dump its cargo in or out. So for now, don't worry about these other processes of endocytosis. If the molecule is small, basically it can either diffuse through the cell membrane or through a protein channel, right? Or it can be actively pushed across the membrane. Bear in mind that generally speaking, non-polar things will move through the cell membrane without a tunnel, whereas polar things will generally need a tunnel. They do note here that because of the force of osmosis, water can move through a membrane to some extent over a long period of time, even though the inside of the membrane is fat soluble and tends to repel water. I don't love this, so I'm going to say mostly think of water as if you want to move a large amount of it because it's polar, you need some kind of protein channel to allow it to move. And the example I gave you in class is an aquaporin. So in any part of your body where you need lots and lots of water to move, you'd need to put channel proteins that allow water to flow. And those are called aquaporins. Yes, it can move somewhat over time, even without aquaporins, but that's a little bit of a thing that I'm not going to ask you to worry about as much, because it makes the whole polar versus non-polar thing more confusing. And again, the reason there is the force of osmosis, but that's something that we'll study in more detail later. So I'd say, again, don't worry about that. Really what we're worried about is non-polar stuff that is small, can go right through the cell membrane. So for example, like CO2 is a small and non-polar molecule, so that can go right through the membrane. Or for example, O2 is a small and non-polar molecule, so that can go right through the membrane. Polar small molecules, say things like H2O, generally speaking, say things like ions like sodium or chloride that are charged. Those things need channel proteins to let them flow. Okay, and then large things need to be coated in membrane, such as like in the example of a vesicle, in order to move in or out of the cell. So I hope that pretty much clarifies how a cell membrane functions to separate inside from outside and what can move across it. Again, keep in mind, if we think about 6-PPD, that tire chemical, it is relatively small. And it is, in fact, it's smaller than like a steroid hormone, and it is non-polar. So we expect that 6-PPD probably could diffuse right through a cell membrane, that it would not be hard for it to do that. And that is why if it rains around here and all of the little microparticles from tires wash into the bay that have built up through our dry season especially, and they contain this 6-PPD stuff, that is why it potentially soaks right through the gut lining and the cell membranes of unsuspecting fish who might consume those microparticles by accident. The 6-PPD can just diffuse right through their cell membranes and have a lethal effect in the case of species like coho salmon. So hopefully that's an immediate application of the concept, which is what I'm always going for here. Now, one final word on, what kinds of things you might look for in an extracellular environment and how we denote that things are dissolved in water. So two really common things to test for in aqueous environments, basically in hydrology and water testing, are nitrate and phosphate. You see their formulas up in the top right. They're both ions. They have charges, NO3- and PO4-3-. They have multiple atoms that form them. So there's 1 nitrogen and 3 oxygens in nitrate, 1 phosphorus and 4 oxygens in phosphate. When they are dissolved in water, we put this little AQ next to them in parentheses. And that means, as I've noted here on the slide, that they are aqueous, which means that they're dissolved in water. They're surrounded by water, okay? We test for these things because, again, to help you to tie things together here, living things are made of BONC, which is phosphorus, hydrogen, oxygen, nitrogen, carbon. And you learned already the photosynthesis equation, CO2 plus H2O in the presence of light gives you glucose. C6H12O6 is the formula for glucose, and oxygen, okay? That is the formula for photosynthesis. Notice that means that as long as there are plants and algae and phytoplankton, things that can do photosynthesis on the Earth, the C and the H and the O, that is the basis of all the food chains and all of the organisms on the planet, therefore, those are basically taken care of as long as there's photosynthesis. And if there's no photosynthesis, there's no life on Earth, at least not as we know it. So the fair question is, where then do we get the other 2 main atoms, the other 2 main elements that living things are made of? And the answer is we get them from aqueous environments. We get them from absorbing them from the water or from the soil, for example, as well. And generally it's plants that are doing that, or other photosynthetic organisms. They soak these in, they add them to their molecules, they build the different molecules living things are made of. And then if someone like a human comes along and eats those plants or eats something that ate those plants, then we steal away the FONC from those other organisms. So that's why the typical water quality monitoring might look at nitrate and phosphate levels, because those are pretty critical to living things. Photosynthesis will always provide you with H, O, and C, but the P and the N have to come from sources like phosphate and nitrate. Okay. Now, what do you think? Quick quiz, immediate application. Pause the video if you need to think about this. Could these pass through a plasma membrane? And if so, how would they pass through? Would it be by simple diffusion? Would they need a protein channel or channel protein? Or would they do it by being coated in cell membrane, being coated in phospholipids, right? And being therefore a vesicle that could be transported across. Okay, hopefully you're ready for the answer. The answer, of course, would be that you need a channel protein for these. They're quite small. They're just ions, right? And ions being small but charged, that means they're water soluble. They do not go across the phospholipid bilayer very easily. They don't go across the cell membrane very easily because the phospholipid tails repel them. So if you want these to come through in any appreciable amount, you need some kind of channel protein in order to make that happen. So indeed, channel protein, channel protein would be the answer to this question. So yes, they could pass through as long as there's a channel protein that allows them to move. And you can bet that if you looked at the cell membranes of algae or of plants, like their root tissue, like the roots of a tree, if you looked at the cells that make up those roots, you'd find channel proteins that absorb phosphate and nitrate, absolutely for certain. Okay. One final thing in this video, just to point out to you, there are different ways of talking about how things are concentrated. Okay, and I think most of you are used to thinking about like percents. Cent means 100, like there are 100 cents in a dollar. In our language, English, it's not so clear that this is the case, but like for example, I learned a little bit of Italian when I was in college, and I really liked the fact that cento is how you say 100. So if you want to say 100%, you say cento per cento, which literally means 100 per 100. And you're like, oh, that makes sense. Like 50% would be 50 per 100. So half of them, 50 out of 100. You can technically convert percent to per anything. So you could do per thousand. And there we'd call it PPT, or parts per thousand. Of course, that's just the percent times 10. Or you could think about parts per million, which would be technically percent times 10,000. Okay. For a lot of pollutants, you measure them in parts per million because they get pretty diluted when they're out in the environment. Their concentration goes down because they get washed into these giant bodies of water like the bay. So they're not very concentrated. They're spread out. Another way to think about concentration of something that is either in a cell or outside of a cell is in the amount of mass per volume, right? So something like grams per milliliter. If I took a milliliter of water, how many grams of that thing would I find? So you might talk about, say, salt concentration that way. Or you could talk about it in parts per thousand, or percent, or parts per million. So depending on the concentrations we need to discuss, if they're in the environment or if they're inside a living thing, we might use different versions of these to talk about them. And often researchers will kind of agree for a certain pollutant, they'll always use ppm because that tends to be the most useful way to think about it. So just being able to kind of think about the fact that there are different ways to talk about how things are concentrated is a good idea. When I wrote these slides, I thought we would probably talk about molarity, and I've paused on that because I don't want to overdo it. Molarity is a fairly complex chemistry concept because it requires you to think about a quantity called a mole. If you're interested and you want to look that up and you want to talk to me about it, great. For now, I'm going to not talk about molarity in class, so ignore that we will learn about it next week because I don't plan on talking about it in this unit. I think it would be too early to get into that, and I don't want to overwhelm people. So for now, my main message is when we're talking about things inside a cell or outside of a cell, we'll often talk about their concentration, and there are different ways of talking about concentration. I just want you to be aware of that and to see some common ways of talking about the concentration of something. Okay. I'm looking forward to working on the lab with you as soon as our soil sieves get here. Remember that basically what we are doing is we are sifting things apart in the soil samples that we took. We're sifting them apart by their size. And then we're going to look at the smallest stuff under a microscope and see if we can classify it as being, human in origin or, non-human in origin, therefore like natural. And hopefully we'll be able to do as the researchers in the paper did. We are using the same equipment as them, so hopefully we can do as well as they did at identifying which microparticles appear to have come from automobile tires, little bits of rubber that have washed off of the road and into those bioswales, those rain gardens that are along the corner of Almaden and Park. All right, that's it for this video. Looking forward to talking to you more about cell organelles in the next one. I hope you got the big themes here are the structure of a membrane and the ways in which it functions to separate in from out and what gets across it and how. And a little bit about concentration of things that are outside or inside of cells, how we would measure that and talk about that. You'll notice in my next video, just to preview it, what I'm going to do now is take the cell membrane as an example of one organelle, one cell part that I've talked about quite a lot. And then I'm going to give you a quick overview of many of the other cell parts and how to think about them not only as individual parts of the cell that can be described in the same way I've described the cell membrane, so made of certain molecules, function to do certain things, but they can also be described together as functioning together to make the cell work overall. So we'll get into that in the next video, and I'm looking forward to it. Thanks, everybody.